just go over the amino acids, and I neglected to do this because most of these you should be familiar with or have heard of. Okay. Is that out of focus? Is it just me, or is it out of focus? Is it? Okay, maybe not. Anyway, so if we start at the top, I'm just going to mention a few of them. Glycine you've probably heard of, leucine, alanine, tryptophan you've probably heard. You know, tryptophan is the mythical compound that's in turkey that's supposed to make you sleep. You know, tryptophan is a precursor of melatonin, I think, something like that. So most of these are common. And glutamate or glutamine, you may have heard of that. You've probably heard of most of these. They're pretty common. And we wanted to pick up today just a little bit of review on the structure. So I added also a link to Khan Academy. How many of you are familiar with Khan Academy? It should be, there's a page on it. There's a page about reviewing organic chemistry, and then in the protein part, there's a link to Khan Academy on the four levels of structure. You know, Khan Academy is fantastic to learn anything, basically. And so I put a link up to like a 20-minute or 15-minute discussion of the 4 levels of protein structure. And we want to pick up here. And I want to make a very, very important point about energy and work. And you've all seen these before, and that, you know, this notion of energy exists, and work is a real thing. And that if I take my sucrose molecule and I split it using an enzyme, that I have to put energy in that system to basically warp the bond between the glucose and the fructose, and of course add water to it and split it apart. And we'll go into detail about how plants absorb light energy and heat and movement. If I take a beaker of sucrose again, I'm going to always try to use sucrose, and I boil it, you know, put sucrose in water and boil it, then I'm going to break down more of the sucrose to glucose and fructose, just because of the heat causing an increase in movement, and the molecules are going to hit each other, and they're going to hit each other sometimes in such a way that there'll be enough energy to split it apart. You know, nothing fancy. Or I can take my beaker of sucrose at room temperature, and I can throw in an enzyme invertase, come back in a short period of time, and all the sucrose has been converted into glucose and fructose. Okay? So this is what an enzyme does. It lowers the amount of energy needed for this reaction to occur. That's what a catalyst does. Another very, very important point is, for the most part, well, in actuality, an enzyme is not destroyed by the reaction. So a very small amount of enzyme does a lot, because it can just keep doing it over and over and over again. The only time an enzyme goes away in a cell is because there's always all these other enzymes called proteases. And what does a protease do? Breaks down proteins. So you have proteases that will eat the enzymes, recycle the amino acids so they can be used again. So mostly we're looking at the first 3 there you should be familiar with, and then the 4th one we're looking at the result of several steps. And remember, I put up that pathway last time where every single step is catalyzed by one enzyme, and in most cases, that one enzyme is composed of more than one polypeptide chain. So most proteins, enzymes have quaternary structures. And there are active enzymes that have 40 or more polypeptides that have to be arranged in that absolutely perfect three-dimensional shape for them to work. Okay? It's actually pretty amazing, you know. And let me give you the enzyme that I know most about is an enzyme called alcohol dehydrogenase, which is a very important enzyme that allows plants to survive flooding. ADH actually in plants produces alcohol. Okay? And of course we have that enzyme too. And plants and humans, we don't have one ADH, we have several. Several different versions of the same enzyme. And they may have different pH optima. There may be one that's expressed in root tissue, and then a different one is expressed in the shoot tissue. So there's just this layer and layer and layer of complexity. Okay? And we'll talk about those reactions, and we'll talk about metabolism. Do we have any questions about this? Okay, let's talk about how we're going to start talking about kinetics. And so we have to first talk about how we describe what happens here. And I'm going to go over this about 10 different ways. So we have a catalyst, which is the enzyme. And that we have this enzyme that's very, very specific. So invertase will only see sucrose. And what I mean by seeing, think about you've got this massively big three-dimensional structure, and you've got negatives and positives and different side groups coming out. And what happens is the sucrose has to bump into it the right way. And because of the interactions, and it's called the catalytic site, and I'll show you that in a bit, the sucrose has to bump into it at just the right way for the enzyme to grab a hold of it. Okay, so when I say C, what I mean it has an anti-sucrose configuration, if you want to do it that way. Okay? So we described something there at the top where we have the enzyme, which is E, and the substrate. And then we have a bidirectional reaction where we have an enzyme and substrate complex. Okay? So the enzyme can sort of grab sucrose and something happens and the sucrose comes out. Nothing happens. So we form this enzyme substrate complex, and maybe it doesn't do anything. It spits the sucrose out. No reaction. Or we do have the other case where the enzyme grabs the sucrose, splits it apart, and we have then the enzyme and the product. Then, I have this on here, but the important thing is the enzyme goes back there. Okay? And it does the same thing over and over and over and over and over and over again. Incredibly fast. You've got invertase in your mouth. You've got amylase. Amylase breaks down starch in your mouth. So you take a sip of Coke, and you already have most of the sucrose converted to reducing sugars, glucose and fructose, by the time it gets into your stomach. Okay? So you start breaking down things really fast. Now here's the cartoon. You've probably seen this in physics. You know, and what this cartoon is trying to show, and energy of activation didn't come off right, is that we have to put energy in here for something to happen. And in this case, the energy is used to bring it up to this peak, and it can fall back down, but in most cases actually it goes ahead and releases the energy, and the energy now is used for this to occur. Okay, the energy doesn't disappear. The energy does not, energy does not disappear. And so it breaks the bond, You grab your sucrose molecule and you split it apart. I'm being very simplistic because, you've got to add water to it and that kind of stuff. But you see this cartoon a lot. The classic one in physics, of course, is a waterfall. And you have the windmill, or not the windmill, the water wheel. And you're using that energy. It's actually removing energy from the water and using that energy to turn the water wheel. Okay? So, the energy actually is used. It's real. Now, here is the classic cartoon or graph of a reaction. Now, the notion is that every reaction will occur and that an enzyme just speeds it up. Well, that's BS. There's a lot of reactions that will not occur without enzymes. But chemically, if you go back to Chem 101, then a catalyst just speeds up a reaction, right? Well, in reality, there are a lot of reactions that are not going to occur unless there are one or more enzymes involved. So if we look at that top peak, we're looking at the left is free energy, and at the bottom is progress of reaction. You need to write this down. Do not confuse this with a V versus S graph, which we've not talked about yet. Okay? We'll talk about that in a bit. And what this shows you, if you look at that energy without an enzyme, is the amount of free energy that must be put into the system. So we're reducing the amount of energy that's needed for this reaction to occur. So if I boil a sucrose solution, I'm putting energy into that system by increasing the Brownian movement. Everybody knows what Brownian movement is. It's molecules hitting into each other, just due to thermal agitation. So I'm going to increase the amount of random breakage of sucrose that's going to occur. Now, if I put an enzyme in there, it's just going to go snap, and it's going to go incredibly fast. Where does energy go? The energy is, to use an analogy, if you grab that sucrose molecule and twist it to break it, that requires what requires energy. Okay, and then there's some reactions that occur that then will release a little energy. In biological systems, okay, and this in biology and physics, is you're trying to conserve some of that energy. And you produce ATP, maybe. Okay? So, in a lot of these reactions, which we'll talk about in metabolism, you're using the energy. You have something that moves a phosphate to an ADP to make an ATP. Now I have a ATP, which is a form of energy, then I can move around in the cell and do something else with. Almost everything we're talking about, when it gets down to it, is oxidation and reduction reactions. And moving electrons around and conserving as much energy as we can, which is different than a physical process. So we're geared up to be as efficient as we possibly can with using energy. Okay. Yes, sir.
So the reason that energy is less with enzymes is because it's trying to conserve energy.
The reason why it's less, well, yeah, The reason, exactly, I mean, the evolutionary reason is because we want to do things with as little energy as possible. And so the enzyme, to use the analogy, it grabs the sucrose and bends the sucrose, so now it's much easier for that bond to break, as opposed to having a bunch of sucrose molecules in water banging into each other. Okay? And again, theoretically, if I were to set a container of sucrose here, keep anything from growing in it so it's completely non-biological, then eventually it would be almost all glucose and fructose. Okay. Invertase is not bidirectional. Invertase will not take a glucose and a fructose and stick it together. That's never going to happen. Even though theoretically everything's reversible. You know, that's never going to happen. Now, a physical biochemist would just, hand me my head if I said something like that. But fortunately, this isn't a physical biochemistry course, which was probably my worst nightmare ever. And this, I'm just putting both of them on here just again to show you that you have some energy going in here. These cartoons are trying to describe energy, which is really, really hard to sort of wrap. It's hard for me to wrap my head around it. Okay, questions? Let me be sure I'm still recording this. Okay, now let's talk about an enzyme, carbonic anhydrase. And in this case, the carbonic anhydrase reaction produces carbonic acid, so it uses CO2 and water. And again, we're seeing basically the same thing. And if you look at CO2 and water, then you have some release of energy over on the right, and we have this thing called activation energy. And if you notice with the enzyme present, then the activation energy of activation is less. So you're taking CO2 plus H2O, and you're ending up, what you're doing is taking the enzyme grabs, and this is a fairly complex enzyme, it grabs these two molecules and sticks them together. Now that's a very simplistic explanation for a lot of steps that occur. The electrons move here through a series of steps, and the CO2 has to have a certain configuration. But what happens is it makes a reaction occur that usually will not occur. If I take water and I bubble CO2 through it, I'll get a bit of carbonic acid. Not a lot, but I'll get some. I mean, that reaction will just happen a little bit. And some people, if they're really, really, really concerned about the water and their pH, is they actually have, when they distill water, they have to put it in an environment that has no CO2 in it so that there's no solubility of the CO2 in the water. Okay? And that's really low. But if you're doing something where you have to be really particular about your water, then you actually have a container that scrubs the CO2. I don't know anyone around here that does that. I'm sure there are people on campus that have to do that. Now, in addition to what enzymes are, which are proteins, polypeptides, and in most cases a bunch of polypeptides, we need metals. These are called cofactors, and we have inorganic metals, which are metals rather, and then we have organic. So the reason you need to take vitamin B12 is so that some of your enzymes will work. The reason plants need minerals is so that their enzymes will work. And of course, other things like we know chlorophyll needs iron for the chlorophyll molecule. And again, what we're talking about that may be new in this class is everything is three-dimensional. So the iron has to be in chlorophyll for the chlorophyll to have the right 3D structure to be able to do anything. Okay? So we have these cofactors, and there are other things but that's why plants and animals need vitamins. Now, what this is a cartoon of is, I can't remember the name of the enzyme now. But this is the relative size of the enzyme and the substrate. So you see in the cartoon there, you can see you've got some sheets and you've got a couple of helices. And then that little white thing there, that little round thing, is the substrate. So to give you an idea of the environment, this fits in here because of all of the side groups on the amino acids in what is called the active site. Okay? And the active site is where the substrate binds. So if I genetically engineer an invertase, the active site has to be almost identical to any invertase in the world. Otherwise, it wouldn't see sucrose. So I can make it a little more efficient. And I want to spend a minute here because this is what a lot of molecular biology is aiming at, is to make the enzyme see the substrate either better or not. Make it not see it as well. And when I say see, I'm talking about all those three-dimensional side groups, those R groups are sticking out. So like Dr. Kyle in our department, he will go in and he'll take a gene and he will change one amino acid at a time in the active site and see how that changes the activity of he works on transcriptional factors. See how that changes how well RNA will be made or DNA will be made. And it's not a hard thing to do. We can go in now and we can, in Arabidopsis, in a plant, sort of a plant anyway, and literally change one amino acid at a time. That's also a reason why he works about 100 hours a week, plus his students. Takes some time, but you can do it.
What makes it bind a specific structure inside of that?
I'm sorry, I didn't hear you.
What makes the substrate bind active?
What makes it bind is that you've got all these protein chains, but it's the side groups. So let's say I have a molecule that's half hydrophobic and half hydrophilic. Then that means that active site, the hydrophobic is going to bind to it's going to be associated with the hydrophobic area, and a hydrophilic is going to be associated with the hydrophilic area. Again, I'm being very simple, but you can imagine you've got negative charges and positive charges, and you've got large subunits, large side chains, and so all that's going to make that phone cord. Again, the phone cord has to be folded up a very specific way every time for that enzyme to work. And if it's an invertase, I can't do much to the active site and still make it be an invertase. I can change it maybe a little, but I can't. If I change all the amino acids, it's no longer an invertase. So all of the chains in the primary structure, all the secondary are responsible for how this thing looks in 3D. And then that very specific site is where the substrate binds. And you're going to see today or maybe Friday that in addition to where the substrate binds, most enzymes have sites out here where other things can bind. And if I bind something here, I may change the way the substrate looks, and the substrate doesn't bind as well. And we're going to talk about that today, probably. Now, let's talk about a couple of ways that we think enzymes work. An induced fit is sort of like the enzyme's a little bit open. The sucrose bumps into it the wrong way, nothing happens. Sucrose bumps into it the wrong way, nothing happens. Sucrose bumps in it just the right way, and the enzyme moves a little bit to grab a hold of the sucrose. So what that is called is the induced fit, because when the substrate binds in the exact, absolutely correct way, the enzyme sort of grabs a hold of it. Okay, and that's one way. And then that then becomes, and when I say it grabs it, it means the enzyme literally moves a little bit, which makes it fit better. It grabs it. You know, like your hand is open, and the substrate keeps the wrong way, you know, and then all of a sudden it's just right, and your hand then will clasp around the substrate. The protein literally changes its three-dimensional shape a little bit, you know. And of course that's going to help that reaction go to completion. Now, here's the cartoon. So what you see is these shapes here, and I want to show you, this is sort of like, again, this is a cartoon. You have this sort of, this thicker or more of a bow, and when it binds, then this part then changes, okay? And then the products are produced, and then the products are released, and now if you notice, we're going back to the original shape that was over here. So this cartoon's trying to show you the changes at the end, so enzyme actually undergoes. It's sort of, you know, shallow here, then it gets thick, then it releases, okay? Now, kinetics. This is something that we're going to be talking about now for a while. And kinetics is how we describe the activity of an enzyme. And below there are listed some factors that can alter an enzyme activity. Temperature, of course, salt content, pH, other molecules, could be other enzymes, could be all of a sudden if you have a lot of increase in proteases, your enzyme activity is going to go down. And of course, the availability of those cofactors. So if I see a plant that's yellow, and by the way, I think it's wrong to memorize mineral nutrition deficiencies. That's really dumb because you could get them wrong. But, you know, if it's yellow, I'm going to say, okay, it's probably a nitrogen deficiency. Why does a plant need nitrogen? Well, a plant needs nitrogen to make amino acids, which makes proteins, which do all these reactions. So if there's no nitrogen, the protein levels are low. The plant can't do much of anything. Okay? So, again, that's why plants need minerals and vitamins. Okay, here's a classic V versus S curve. Anybody here had biochemistry? Biochem, yeah. Okay, we're not going to go through the derivation, so, but what we see here is a very interesting curve. And we look at the substrate concentration along the x-axis, and we look at the reaction rate. And if I see this, I know that this reaction is mediated by an enzyme. Okay? Now let me also tell you that this is also, if I were to grow plants in different concentrations of iron, and iron being the substrate, and I looked at uptake of iron into the plant, I would get this same curve. That's because iron has to be carried by a protein across the membranes. Okay? Same thing. So why do we get this shape? Why doesn't it just keep going up?
Because at some point, you could just pack it full
Right.
It's not going to benefit anyone really.
Right. What we see is, and what I like to use as an analogy is, let's say out of our loading dock back here, let's say we got a guy moving boxes from a truck. Okay, he takes a box out and moves it inside. Everybody following what I'm saying? Truck pulls up, it has one box. How many boxes can he move in a minute? Or say 10 minutes, how many boxes? If it has one box in it, how many can he move in 10 minutes? One. Next truck comes in, has two boxes. Maybe he can move both of them in a minute. Next truck comes in, let's say it has 10 boxes. Maybe he can move 5 in a minute. Next truck comes in, it's got 1,000 boxes. How many boxes can he move in a minute? 5. So the amount of the enzyme is going to determine how many boxes, the number of enzyme molecules, it's going to determine how many boxes can be moved, okay? So we saturate the system. So it comes in with 1,000,000 boxes. How many boxes can he move? 5. Whatever. 5, 6, 7, 4. Never, depends on whatever. So when we see this curve, we know that usually this is mediated, enzyme mediated. And there's some terms that you will need to know. And these terms that we actually calculate for each enzyme because it describes what the enzyme can do. Let's start at the top, which is Vmax, which is the maximum velocity. Okay? What units do I have to add for it to be a velocity term? What's the units? For it to be velocity, speed. It's got to be something per unit of time. So this protein will do 5 boxes per minute per person. So it's per protein. So 5 boxes per minute. And then we look at another term, and these are, and we're not going through the derivation, but we're also interested in 1/2 the maximum velocity. Okay? And the reason we're interested in 1/2 maximum velocity, there's a lot of reasons. There's a lot of mechanical reasons. But it helps us describe the enzyme. And at 1/2 maximum velocity, what we like to determine is something called the concentration, Km. And Km is the concentration at 1/2 maximum velocity. So the Km has the terms, would be 2.5 boxes, whatever. So Km is concentration. Okay? Vmax is velocity, V is velocity, and Km is concentration. And we just draw this curve. Okay? Now you'll notice the Km is way down here. So because remember, the way this curve is shaped, at 1/2 maximum velocity, which we're saying the maximum velocity here is about 32 or 33, so 1/2 maximum velocity is going to be about 17 or 18, and the concentration is low. Okay? So let's say that's what, 500, maybe 500, 400, whatever the concentration. So 400 nanomoles, 400 boxes, whatever, at the same. Okay? Questions? Okay. So let's draw a curve and let's plot this. And we're going to do boxes, so we're going to do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And we're going to do products, which would be boxes inside. Okay, let's go down to 0. How many boxes can this person move if the truck is empty? How many boxes? 0. Makes sense, right? Truck comes in with 1 box. We get a velocity of, let's say, 1 box per minute. Let's say 0, 1, 2, 3, 4, 5, 6, 7, 8. Okay. 1, let's go 2, would be 2. 3, let's say 3. Let's max them out about 4. So, I can't draw. So 4, then we'd start seeing something like this. And you're going to do this multiple times, so sometimes maybe he can only move 3 boxes, you know. And so what you see then is that curve. Okay, not drawn very well. Okay, questions about that? So we're saying saturation doesn't matter. We can increase the number of boxes by increasing the number of people that can move the boxes. But what we're doing is describing the characteristics of a set number of enzyme molecules, a set number of people. Very important because this describes what this enzyme can do. And one interesting thing, excuse me, help me go back, is what we find is that the concentration in the cell of a substrate is right around this Km value for the most part. So what does that mean? Yep. You add a little bit of substrate, the reaction goes up a lot at that point. You reduce a substrate, the reaction goes down a lot, really fast. So during this period right here, you know, you're basically almost, not quite, but almost linear. Increase it a little bit, reaction goes up fast. Decrease it, it goes down. So it stands to reason that at the cell level, usually the concentration of the substrate is going to be not 1,000 times above the Km. It's going to be twice the Km or a little bit below the Km, because that's the part of the reaction that is really that we can speed up or slow down, okay? And with that, Friday, we will cover inhibitors. Any questions? Take a look at this. This is a fairly complex phenomena, okay? Okay. You guys get out 5 minutes early. I'm just too liberal with